We have studied that DNA formed after replication consists of two strands, one that is received from the parent DNA and the other that is newly synthesized. This type of replication is called semi-conservative replication. The Meselson and Stahl provided an evidence for semi-conservative replication of the DNA molecule. They grew E. coli cells for several generations on a medium containing heavy isotope of nitrogen, that is N15. Then they isolated the first bacterial sample and extracted the DNA, assuming that all the DNA was labeled with N15. A concentrated solution of cesium chloride was then added to this sample and was centrifuged at a high speed. This test tube was labeled as 1. Simultaneously, some of the bacteria were transferred to a medium containing lighter isotope of nitrogen, that is N14. Samples from this medium were taken at regular intervals of 20 minutes and were labeled as test tube 2, 3 and 4. These samples were then centrifuged at high speed with cesium chloride. After analysis of test tube 1, 2, 3 and 4, it was observed that test tube 1 contains DNA with 100% heavy isotope, that is N15, since it has been grown in only N15 medium. Test tube 2 contains first generation DNA with 100% intermediate density DNA, that is both N15 and N14, grown in N14 medium. Test tube 3, having second generation in N14 medium, contains equal amount of intermediate density DNA and DNA with light isotope, that is N14. Test tube 4, having third generation in N14 medium, containing 75% DNA with light isotope as compared to 25% DNA with heavy isotope. So, on the basis of the evidence provided by this experiment, it was concluded that process of replication is semi-conservative.